welcome to this lesson for Advanced Higher Biology. We are looking at Unit 1, Key Area 1. We're in Laboratory Techniques. And today we are going to focus on separation techniques, what these are and why it is that we need to carry them out. So for this section, what we would expect you to know is understanding how the different substances can be separated from one another. You need to be able to describe this process of centrifugation, explain the basic processes of paper and thin layer chromatography, describe what we mean by affinity chromatography, describe the principles of gel electrophoresis, native gels and SDH, and explain how proteins can be separated from each other using their pH or IEP. So to consider separation techniques in their entirety, when we are carrying out any kind of practical work in the lab, sometimes we need to separate different substances from each other. And depending on what substances are involved, this could be done on the basis of their density, their solubility, their shape, their size, or their overall charge. So we're going to take each of these properties in turn and look at the different techniques we can use in order to separate them out when we're using those. To begin with, we will take density. So density, we will use a centrifuge usually to separate things out on the basis of their density. A centrifuge is essentially a piece of lab equipment and it will spin tubes of liquid incredibly fast. And as it spins, the more dense components of a mixture will move further down the tube than the less dense components. So depending on what it is we're wanting to separate and precisely the particular substance that we need in order to centrifuge it, we may use a different level of spin. It's important to realise when we're looking at this diagram here, although our molecules are a different size, that's not always the case in terms of their density. So this has been a simplified diagram, but just to clear up that potential misconception there, larger items are not necessarily more dense. We need to consider the size of our molecules at a different level. So we're thinking about their density, not just their physical size. It just so happens that in this particular diagram, the larger ones are more dense. So if we just wanted to extract or remove this green one, for example, maybe we want the rest of the substance and we're just wanting to get rid of this. Now that we could put it on low spin speed, and that would leave us with the more dense material down at the bottom, and we'd be able to use the material that's still dissolved, that's still in the solution through the rest of the test tube. Obviously, if that wasn't the case, if we actually wanted to keep, say for example, this nice little black one down here, then what we could do is we could use a very high speed spin and we could separate out individually. Now, when we are looking at our centrifugation or the process of centrifugation, we've got a few different terms to think about. So when we look at centrifugation, the section that is dense, this section here, which is pulled out, is referred to as the pellet. So our pellet would be that section. On the other hand, our other substance up here is what we refer to as the supernatant. And as I said, depending on what substance it is you're separating, you may wish to use either the pellet or the supernatant, depending on what it is that you're actually trying to separate and what it is you're looking for, what it is you're trying to use. Now we can also use the process of chromatography in order to separate substances out by their solubility. And we can use either paper or thin layer chromatography for separating different substances, and it tends to be things like amino acids or sugars that we would use this process to separate. Now, both thin layer and paper chromatography are actually very similar, so as we're describing this, consider it could be on either one, and we'll mention the differences about them just towards the end. Now, when we're carrying out chromatography, we would normally use various materials, often it's separate dyes that we might be separating, and what we're going to do is we're going to use particular paper, chromatography paper, We're going to draw a start line on it with a pencil usually, so it doesn't affect the chromatogram. And we would usually take a small section of our substance that we're wanting to separate, and we would put a spot of that on towards the base of our chromatography paper. We would then put a solvent into our beaker, and that solvent is going to pull the different constituents of the various dyes up the chromatogram. And the different solubility of those materials that are being tested, so whatever it is that makes up our dye here, is going to result in different speeds and therefore give us different substances. So over time, as we look at our chromatogram, what you would expect to see is the solvent is slowly going to move up the way, so eventually it will come towards the top of our chromatography paper. It'll be absorbed up our chromatography paper. As it does that, what's happening is our actual process is also taking up our different materials depending on what it is that they've actually got within them, and the different processes, the different parts, that they've got in them. And where that's the case, for example, what we'd look at on this one is we might expect to see some sort of smudges moving up here, and that would give us our components of this dye, and we'd be able to compare it to our other ones which have been along here, and that starts to tell us the sorts of components that are actually present within the original dye. We're able to start comparing them and seeing if they're at all similar to our original, and seeing what's present in those ones. So what you might see overall towards the ends would be ones that looked similar to this one. You might start seeing similar bands appearing in similar areas depending on the original dye, and that would allow you to really identify or have separated out your individual parts of your dye or your material. It's often something that may have been done when you looked at photosynthesis. You've maybe extracted the chlorophyll and looked at the range of different chlorophylls and the different colours that are present in them. And when we compare paper and thin layer chromatography, what we need to think about is the fact that our paper, it tends to be made of cellulose, that's highly hydrophilic, which means it is water loving, and the solvent tends to have hydrophobic properties, and that's what allows our solvent to move up our chromatography paper. And within our solvent, obviously, each solute is going to be able to travel up the paper along with that, depending on its different solubility in the solvent. Now, very similar solvent properties are essentially used in our thin layer chromatography, but in this case, rather than being made of paper, the thin layer is usually made of a thin silica gel, which would be used instead. Now, we then have to consider how we may separate different substances from their shape. In this case, it would usually be affinity chromatography, and it tends to involve a protein rather than any other type of mixture. So it might be a mixture of proteins, and we're looking to separate one specific soluble protein from this mixture. What we're going to do is we're going to have an antibody or a ligand, and these are things which are specific to binding to the protein that we would like. So this immobilized molecule, which is complementary to our target protein, is placed within, so that's it here, that's placed within this particular column. It's in a gel-like substance, and it's packed into a small plastic column. So we've got this immobilized molecule attached to that gel, and then what we're going to do is we're going to put our mixture of proteins, we're going to pour it through the column. And as we pour it through the column, only the protein that has got this high affinity to this antibody or ligand will bind onto the column. So it's only that one that's going to stick. The other ones will not bind. They will in fact be washed out as they have a weaker affinity. Now, again, depending on which protein it is we want or what we want within our solution, there are a couple of different stages we could do at this point. So if we wanted to, say, get rid of that bound protein, then we would just take our mixture, and that would be the mixture that we would then go ahead and use without the protein that we didn't want. On the other hand, It might be the case that actually we did want this particular protein, that was our target protein, in which case we will then wash the column out. That's usually done with a buffer solution, and it's usually got a different pH to what we started with. That basically changes how much the protein wants to bind to the ligand, and it will wash the protein out of our column. The alternative would be to use a solution which would have free ligand molecules, so the solution itself would contain those ligand molecules and the protein would bind to it instead and be washed out. So again, it really depends on what it is we're wanting to do, whether we're wanting to use the target protein by itself or whether we're wanting to separate it out completely. Now, our last process that we're going to consider is that of gel electrophoresis, and this can be used to separate molecules by either size or charge. Essentially, the process is going to use a current, and that flows through a buffer and will either separate out proteins or nucleic acids. As you can see, we've got our electrophoresis tank set up here. It's got a gel within it. Those gels have sample wells where you can put your sample into. We've got positive and negative electrode and a buffer solution, and consequently our samples will move from the negative to the positive electrode depending on their size or depending on their charge. It's mostly commonly seen where we look at separating out DNA in order to carry out DNA fingerprinting, etc. So the charged molecules are going to move through your electric field. First stage is applying our DNA or protein samples to be loaded into the gel. We would then turn on the power source and the fragments are going to migrate through the gel. Different fragments will move at different speeds depending on their size and depending on their charge. So usually your larger and heavier ones are going to stay towards the top end where you've loaded them, and your smaller ones are going to have run further down. We also have gels referred to as STS page gels, and in this case they actually give all of the molecules equally negative charge and denature them, and that separates proteins out by size alone. It is also possible to run what we refer to as native gels, and that's where the protein itself is not denatured before the gel electrophoresis, and that allows us to look at or analyse our proteins when they're in their folded state. And when that's the case, the migration of the proteins through the gel usually depends on the protein's shape, size and charge. Finally, we can consider the isoelectric point, or IEP, of a protein. And when we look at these, this is the point at which a protein has no net charge. So it's the pH of solution that our proteins don't have a particular charge at. Usually, when we look at a protein within a solution, depending on the IEP of the protein, there will either be a majority of positive or negative charges at the surface of the protein. So where our pH of our solution is less than the IEP, we tend to see an overall positive charge around the protein and on the protein's surface. So remember, this is 3D molecules, so we would expect them to be coming out the way as well. Where the pH is greater than the isoelectric point of the protein, we would tend to see overall negative charges surrounding our protein and on its surface. Whereas where our pH is the same as the IEP, or gives our protein its IEP, then the balance of the charge tends to be a bit more even. So we would have an even number of positive and negative charges on the surface of our protein. IEP of our protein, there is an overall neutral charge, and that allows the protein to form a solid and precipitate out of solution. So the solubility drops when the protein reaches the pH at which it's got its IEP. So that technique can be used to separate proteins from one another. For example, if you have a solution buffered to a specific pH, that will allow only proteins with an IEP of that pH to be able to precipitate, to be able to solidify and come out of the solution. It's also possible to take soluble proteins and separate using them, using an electric field and a pH gradient. And the protein would stop migrating through the gel at its IEP in its pH gradient because it's got no net charge. So if we're starting with this mixture of proteins in A and we've got our positive and negative ends of our electrodes, because of that, we've also got a gradiated pH. As a result, our proteins are going to move, obviously our negatively charged proteins are going to move towards the positive end, our positively charged proteins are going to move towards the negative end, resulting in our proteins having moved through the gel and finishing up, stopping there at their IEP. So from that, you should be able to describe what we mean by centrifugation, explain the basic processes of paper and thin layer chromatography, describe what we mean by affinity chromatography, describe the principles of gel electrophoresis, native gels and STS page, and explain how proteins can be separated from each other using either their pH or their IEP. Thank you very much for watching, and please let me know if you do have any questions.